So I'm here today to tell you a little bit about the research that I do and the research that we do here in Macau. So I'm an associate professor, as Sid explained. I'm an associate professor and leader of the Astrobiology unit at the Macau University of Science and Technology. I do come from a microbiology background. I am originally from Portugal. That's where I did my undergraduate studies, did my PhD as part of a joint degree between my institution back home in Portugal and the University of Regensburg in Germany, a very famous center of research of archaea and extreme environments. I've worked in a variety of different institutions and different places, and I am now in Macau. I'm not really sure within the audience if you all heard about Macau before, if you know where Macau is. So Macau is a special administrative region of China. So it's part of the one country, two systems. It's in the delta of the Pearl River, and it's an amazing city. It's a unique combination of modern and ancient buildings, Chinese influences and Portuguese influence. So it's a unique blend, a mixture of different cultures and different people, which makes for a really nice place to live in. I'm working at Macau University of Science and Technology. I moved here a couple of months ago and joined the State Key Laboratory of Lunar and Planetary Sciences, which is also a recent addition to the university. We have a campus with an amazing location. We're right next to the big casinos that you might have heard about. We benefit also from a very privileged link with the China National Space Administration. And the university has been doing really well. It's still rather recent. It's just turned 20, but it's been doing really well in the rankings. It was listed. This is data from last week. We were just recently recognized as the 38th best young university in the world by the Times Higher Education ranking, which is something that we're really proud about. Regarding research topics, so I'm the leader of the astrobiology unit. We do research in a variety of different fields. Our main focus is on terrestrial analog sites, particularly focusing on marine and extreme environments. And we explore the limits of life, both from bacteria, archaea, and fungi, and focusing on biodiversity, ecology, and a little bit of bioprospection also. We work with geomicrobiology, the interaction between microbes and minerals, which is an exciting new field. And I also have a colleague as part of this unit that leads the geobiology part of research and focuses more on biosignature and microbial preservation. And we also do some work in development of new techniques, new tools. And of course, we're directly linked with astrobiology research and planetary protection. An important thing that I wanted to mention, because otherwise I might forget this at the end of the talk, we are currently recruiting. We are looking for new faculty. So as I explained, the State Key Lab is new. It's expanding quite quickly. We're currently recruiting new faculty for the different research lines in the department. We're also constantly getting new postdocs. We're very keen on getting additional PhD students and master's students. So if you're interested in some of the things that I discussed here today, or just curious and want to know more about what we do here, don't hesitate and just shoot me an email, ask me questions about it, okay? Even if it's not linked with astrobiology, I'll forward it to the right contact person in the department. Okay, so as the first part of this talk, I'm going to focus a little bit on brines. And before I start with that, I need to give a little bit more context on microbiology. So microbiology is, as we know it today, is heavily linked with the work of a couple of pioneers, people that did breakthrough research in this field. And of course, the very first person that I need to acknowledge is this guy here, Antonie van Leeuwenhoek. And Antonie van Leeuwenhoek was the father of microbiology. He was the first person to actually visualize microbes, observe microbes, and did the first rudimentary drawings of what he observed. He was a very curious person. His family had a business in textile industry, And they used this special tool, the special gadget that you can see on the right side, that was used to analyze the quality of textiles. And so, because he was very curious, he adapted this gadget and started trying to observe different things that surrounded him. And that's how he was able to identify that we were surrounded by very, very small life forms that we weren't aware of beforehand. Additional researchers that were very important in the development of microbiology, we have Robert Koch, of course, and particularly Fanny Hess. Fanny Hess worked directly with Robert Koch, and she's not normally acknowledged for this. She had a very important contribution in the development of agar plates. These typical Petri dishes that you've likely seen in photos and TV if you don't work with microbiology. She played a very important role in the use of agar to make these Petri dishes. Before she suggested the use of this compound to make solid media to grow microbes on, people were relying on slices of potatoes, believe it or not, to try and grow microbes and isolate them and get pure isolated colonies and study them a little bit better. So this was a very, very important breakthrough there. And finally, the other pioneer that I need to acknowledge is, of course, Carl Woese. And Carl Woese was a revolutionary researcher. He was the person that suggested the use of genomic information, more specifically the sequence of 16S ribosomal RNA, the gene that codes for 16S ribosomal RNA, as a way of mapping phylogenetic relationships between different organisms. And so he's directly responsible for our view of life and how life is organized and related. Different organisms are related to each other in our planet. And so we now have this typical visualization of diversity in our planet. We have the so-called tree of life with 3 big branches, eukarya, which includes animals, plants, we're also there, fungi are there. Then we have bacteria and archaea, which are both are simpler organisms, and our prokaryotic cells that do not have a defined nucleus. And so this kick-started the use of molecular biology in microbiology. And so given these new tools, we now have a complete view of the world and life's diversity in our planet. We now know that we live in a microbial world, that microbes dominate our planet. And despite the fact that they play this dominant role, we've only been able to study less than 1% so far. So there's still a lot that we don't know. And they're clearly the dominant life forms. The abundance of microbes is mind-boggling. Just to give you a comparison that is normally referred to, the number of bacteria in our oceans clearly and vastly outnumbers estimates on total number of stars in the universe. So that's how massive a role bacteria play in our planet. In addition to that, they even outnumber our own cells in our bodies. Current estimates point to us having 10 times more microbes in our body than our own cells. And they interact with different organisms and have different types of effects, which we're just starting to understand. In addition to that, microbes are incredibly resilient. They're very, very sturdy, and they're easily adaptable, and they're able to cope with different types of conditions. And so what we realize is that different types of extreme conditions that we find in our planet, including several of them that were originally thought to be sterile, that they were too extreme and nothing could survive these conditions, ranging from low pH conditions, and you see there in the upper left corner photo of Rio Tinto in Spain, to high temperature environments, and there you have a photo of Yellowstone, to alkaline conditions, low temperature. Microbes do really well under different types of extreme. Extreme after extreme, we've been able to confirm that microbes live there and do really well under these conditions. And of course, my favorite is high salinity environments. from coastal lagoons to high salinity lakes, salt mines. There's a wide range of high salinity environments that are really interesting. And it is important to note that when we discuss organisms living in extreme environments, we're not just saying that microbes are able to survive in extreme environments. They love these conditions. These are the ideal conditions for certain types of organisms. And without that, they're unable to live. So we need to be a little bit careful with the whole definition of extreme and what we consider extreme, because we do have a human-centric perspective there. And what we consider extreme for them is the normal conditions that they live in. And so when we're discussing organisms that live under extreme environments and thrive under these conditions, we normally call them extremophiles. And so there's a wide range of different extremophiles depending on what type of extreme we're discussing. And specifically when you're looking at high salinity environments like brines, organisms are called halophiles. They love high salinity conditions. And so if you've lived in coastal regions before, or if you had contact with coastal regions in sunny, warm environments, you've probably seen this sort of landscape before. So these are evaporation ponds at a salt production facility. It's a very common way of producing salt. And you see this very characteristic colors, pinkish, reddish, orange coloration that are associated with these high salinity lakes. The reason behind this color has to do with very high density of microbes. Microbes that have specific pigments that induce this color change in water. And so despite the fact that conditions are quite extreme, that the salinity in these sites is quite high, we do have representatives of the three domains of life that are able to cope with these extreme conditions and do really well. So we have animals like Artemia, we have plants like the microalgae Dunaliella, and then we have both bacteria and archaea, including some unusual shaped ones like Haloquadratum. And of course, when I mention, when I discuss and when I say that high salinity environment is extreme and is harsh and it's difficult, it poses some challenges to life. The big challenge there relates with water activity. And so water activity is a way of measuring water that is available for biological functions. So when you have high salt content, the water molecules tend to interact with these salts and they become less available for normal activities in cells. And so that's the big challenge, that water has, despite the fact that you have a liquid environment and you have water with salt, not all water is available for performing biological functions. We do have some striking similarities with high sugar environments, like syrups, for example, different types of candy, honey. And so the challenges are the same. So we do have other types of extremophiles like osmophiles and xerophiles, so organisms that live under high sugar environments and dry environments. And the challenge is the same, making sure that organisms have enough water to be able to perform and to function regularly. And so just to give you an additional component there, given that cell membranes, and this is an image of a plant cell, not of a microbe, but it does give you that sort of perspective. The big issue that you have when you place a cell in a high salinity environment or a hypertonic environment is cells will lose water. So the concentration of solutes inside the cells is usually lower than in environments that have a lot of salt or have a lot of sugar. And so because salts or sugars aren't able to freely cross the biological membrane, but water is, then water will flow from low environment to low concentration environment to high concentration environment to sort of try and dilute it and even things out. And so that's the big challenge there. So how do organisms adapt to high salinity? How do microbes cope with this? There are two different approaches, the salt to end cytoplasm strategy and the organic osmolyte strategy, also known as compatible solute strategy. And so microbes, so the salt-in strategy is quite simple. Organisms for certain organisms to adapt and be able to keep water inside the cells, what they do is they increase the salinity inside the cytoplasm, the salts inside the cell, to sort of match the concentrations that you have outside. So this allows, of course, water to be retained. It does involve significant changes to the machinery of the cells. All types of enzymes, all types of structures need to be able to operate under high salinity conditions. And this is a typical strategy for halophilic archaea. That's the standard, although there are a few cases of bacteria that also do it. The compatible solute strategy is slightly different. Rather than accumulating salt, they accumulate different molecules, organic molecules, that counterbalance the high concentration of salts outside. So it's a way of retaining water inside the cells. And it does give organisms a little bit more flexibility. An additional thing that I wanted to mention that is relevant is the discussion on cosmotropes and chaotropes, which is something that I discussed with Tony in last week's webinar. And I don't want to spend too much time here. It's just important to keep in mind that not all salts are the same. There are different types of salts. And so we can differentiate, we have this range between chaotropes and cosmotropes. And cosmotropes have less disruptive properties than chaotropes. Chaotropes induce more chaos. They have the capability of disrupting normal proteins and normal biological structure. So they have a more detrimental effect on life. Okay, so one specific type of high salinity environment that I'm a big fan of and that I dedicate a big part of my research life on is deep sea brines. So deep sea brines are very unusual environments. They were discovered purely by accident in the late 1950s. A Swedish research vessel was doing a survey of the bottom of the Red Sea, and they collected a sample that had unexpectedly high salinity and very high temperature. And this was something completely unexpected. There was a lot of interest in trying to understand what created this. And we now, follow-up surveys identified several other deeps with similar conditions, deeps that were filled with brines, high salinity water. And so we now understand a little bit better what creates them. So we have the Red Sea is an ocean that is being formed. It's still just a sea, but you have this split between the Arabian and the African tectonic plates. This tectonic rifting creates several isolated depressions at the bottom of the sea. It creates several deeps. And in addition to that, so this explains why the deeps are there, but where does the brine come from? The brine comes from remnants from a prehistoric ocean that existed in this area underneath the whole of the Red Sea. And so this prehistoric ocean dried up and created this thick layer of salt. So underneath the whole of the Red Sea, you have thick layer of salt, not that deep. And so when you have tectonic rift processes, you create cracks in the Earth's crust. And you're exposing the salt layer to contact with regular seawater. And so regular seawater trickles down and reaches the salt layer, dissolves the salt, it becomes heavily enriched in salt. And because you also have associated hydrothermal activity, it resurfaces as warmer water that has a very high salt content. It can go all the way to saturation. You also have an increase in heavy metal concentration, depletion of oxygen, and so there's some physical chemical changes to regular seawater. Because brines, because of their high salt content, brines are very, very dense. And so they tend to stay in deeper areas. And so that's how they eventually fill all of these different deeps that you find in the Red Sea area. We currently know around 25 different ones. And as two extreme examples here, we have Kebra Deep, which is one of the northernmost ones, and it's one of the smallest ones. It is relatively cold. Temperature is close to 24 degrees Celsius. And it's small. It's around 2.5 square kilometers. This is in contrast with Atlantis 2. Atlantis 2 is one of the oldest deep sea brines, and because it has had so much hydrothermal activity and hydrothermal input, temperature is higher. Temperature is currently at around 68 degrees Celsius, but it keeps on increasing. So ever since it's been discovered, temperatures have risen by, I think by around 20 degrees Celsius. So that's quite a lot. And it's a very big brine. Despite the fact that you have this common origin process, they all form the same way and they're linked with this sort of tectonic rift process, we do see some chemical variability. This has to do with local geological settings, okay? Because geological layers are slightly different. Then you have different types of chemical profiles with enrichment of more of specific types of ions in certain brines when compared to others. I could also add that much later, there were some additional discoveries, some additional deep sea brines that were found in the Mediterranean area, and then later also in the Gulf of Mexico. They do come from a similar sort of setting, so tectonic activity associated with an underground layer of salt from prehistoric ocean that dried up. Well, I mentioned before that deep sea brines are, well, the brines are very dense. And because they're so dense, they don't really mix much with regular seawater. This creates this drastic, this very sharp transition zone between conditions in seawater and conditions in the brine. And so this brine seawater interface is a remarkable type of environment where you have transition from low temperatures to high temperatures, from oxygen to anoxic conditions, drastic increase in salinity, and it's a really interesting environment. We do see very marked differences between samples that we collect at different stages of the interface, as you can see from the filters that I have here. I have here some video footage. So we were very lucky in one of the last expeditions that we had, that I participated in. We had an ROV, so we were able to collect video footage. The camera is starting to get into the brine seawater interface. You will see an increase in particle load. So you see this fuzzy layer, and the camera just entered the brine. The accumulation of these particles has to do with the density, the high density of the brine. Particles tend to accumulate at the interface because they're not heavy enough to break through the brine seawater interface. We also see some macrofauna, as you saw in the video. There was, and this is video footage from Kebra Deep. We also were very surprised to find these unusual patches spread across the brine seawater interface, that had this sort of typical reddish coloration, and it seems to be some sort of biofilm. Approaching it with the ROV quickly disrupted it, and we're quite curious to know what this is. Well, we still don't know. It could be that it's some sort of higher density of halophilic archaea, for example, that perhaps tend to have this very bright red pigmentation associated with it. But really interesting environment that we have here. Okay, so how do we study deep sea brines? This is one of the questions that I get on a more regular basis. People ask me, oh, do you go down there? Do you dive? How do you get there? The depth is quite high. The shallowest deep sea brines in the Red Sea, you can find them at around 1,500 meters, but the majority of them usually start at around 2,200 meters. So that's quite deep. We make use of these special gadgets that are commonly used in oceanographical studies. So we have rosette samplers, and they're equipped with these very special type of bottles that are called Niskin bottles that you can see over there. They have some lids in the upper and the lower part, and they have I'll explain how they work in the following slide. And we also make use of things like multi-cores to collect sediment samples. And so Niskin bottles, and they're equipped there in a rosette sampler, they're lowered via a cable and they're connected to the research vessel that we use, to the ship. They have all sorts of sensors, and Niskin bottles are lowered with both lids open, both valves open. And so basically what you have is a hollow tube that is being lowered through the seawater. Once you reach the depth that you're interested in, and you know that because you have exact information on what depth you're at, what's the temperature, what's the salinity, what's the oxygen concentration. Once it reaches the depth that you're interested in, you just trigger the system and it simultaneously closes both valves. So it successfully traps seawater or brine inside the Niskin bottle. And so it's this type of samples that we bring back to the deck, and then we're able to collect samples from several different depths. I was mentioning before the drastic changes that occur at the brine-seawater interface. We were able to collect samples where you see variation within one bottle, within one Niskin bottle. If you take a sample from the bottom part or from the upper part, you have completely different salinity levels. Okay, so what about microbiology? What is it that we know about microbiology of these sites? So there was a lot of interest in the geochemical aspect. Part of it had to do with expectations to find oil, because it was the Middle East and you have high salinity environments. Brines are frequently associated with oil deposits. Regarding microbiology, first studies were were done in the late 1960s, and this is what they found. Nothing. I actually have papers from that period that say conditions are too extreme. No life forms can survive these conditions. No way any type of organism would be able to live down there, because you had high pressure, high salinity, high temperature, high concentration of heavy metals, no oxygen. It was considered too harsh. And so it's understandable. In the 1960s, we didn't have the tools that we have nowadays. And a lot of the microbiological studies were limited just to cultivation, just preparing some Petri dishes and putting your samples in there, spreading them, and see if anything would grow. So there's some limitations to this approach. Of course, nowadays, microbiology makes use of this holistic approach where we collect environmental data to understand what type of environments microbes come from, coupled with molecular-based approaches like metagenomics, for example, where we get a little bit more information of what's the potential, what are the capabilities, and what's the real diversity that we find in nature. And of course, all of this links back to cultivation, to isolating microbes and understanding what they are and what they're capable of. And so first phylogenetic studies in the late 90s and early 2000s brought a big surprise. Contrary to this original view, researchers were able to show that deep sea brines in the Red Sea were filled with life. This microbial life was incredibly diverse, incredibly abundant, and there was a high number of new groups of microbes, completely new to science, and not really closely related to any known microbes on our planet. Work continued in the following decades, including several studies done in deep sea brines of the Mediterranean. And so we now have identified several new groups of microbes that have no close representatives and seem to be specifically adapted. We only find them in deep sea brines. And it's really interesting because some of these groups you find in all deep sea brines. All of the brines in the Red Sea, all of the brines in the Mediterranean, they're always there, and they're not found anywhere else, which raises some interesting issues. Where did they come from? Which is something that we might have some time to discuss it as part of the Q&A session. Okay, what about microbial isolates? What microbes were we able to get from these environments. As I explained, this was the original view. No microbes were, well, it was not possible to isolate any microbes. Part of it had to do with limitations of this technique. And it's not unusual to have this type of result. And we're getting better at this, but we're still lacking in our capabilities of isolating microbes and studying microbes in the lab. So there's this very well documented mismatch between number of microbes that we see in the environment and diversity that is present there when we look at under the microscope, or when we use molecular techniques and we look at DNA that is present in the environment, when you compare with results that you get on plates, where you have a limited number of colonies, limited diversity, and this mismatch is known as the Great Plate Count Anomaly. Okay, there's a variety of factors there. Big part of it has to do with the fact that we might not be using the right conditions, or we might not be using the right techniques. One very important tool to avoid this, or to help us minimize this, is this really handy gadget that I'm going to show a video of, sorry. So this is video footage from a laser microscope, and laser microscopes are used for this very refined technique called optical tweezers. And so something that I'm very happy to report that we're getting one of these for the lab, for our new labs. And so what you have here, this is, I'm sorry for the music. This is a microscopy image, and you see several different cells there. And the cross that you see there in the middle is a laser beam. When you activate the laser beam, you're able to fix one specific cell, one individual cell in position, which is something really cool. And it's not just cool because, well, lasers are cool. It's really cool because it's useful. It's incredibly useful for isolating new microbes, and microbes that do not grow on plates, microbes that we struggle getting in pure culture in the lab. And so the fact that we're able to fix one cell in position allows us to isolate one individual cell from a mixture of different cells. And where we can adapt this to a glass capillary, where we physically remove one individual cell from all the others, and then we can transfer it to different types of media. We can transfer it to growth media, and we can get pure cultures in this way. So it's a really, really very powerful technique, and it's very handy for microbiology of exotic microbes. Okay. Before I give you some examples of some of the microbes that we isolated, that we've been able to get from these deep sea brines, I just wanted to give you a brief overview of different taxonomic ranks. So I think that you're all familiar with the concept of species and the concept of kingdom. I already mentioned the domain concept before. So most of the microbes that I'm going to mention today, that I'm going to mention as the examples in the following slides, are new orders or new phyla, for example. And so this would be equivalent, just to get a better perception, this would be equivalent to finding the very first carnivore or the very first chordate. So that's a massive new discovery in our understanding of life's diversity. So the very first microbe that was isolated from deep sea brines ever was this one, Flexistipe sinusarabici. It represented a new phylum. It is a thermophile, so it does well under high temperature environments, and this fits really nicely with the original environment. This microbe comes from Atlantis II, which is the hottest deep sea brine. Another one is Salinisphera chabanensis. This was the first microbe that I isolated as part of my PhD. Salinisphera chabanensis represents a new order and has some really interesting characteristics, namely the fact that it's very, very flexible, very versatile, which fits really nicely with the fact that it comes from the brine-seawater interface of Chabandy. So where you have this drastic transition of conditions, it seems to do really well under completely different conditions of salinity, temperature, oxygen availability, and this should be a big advantage in that type of challenging environment. We've also analyzed the genome later on, and so the genes, some of the genes that we identified, fit really well with this lifestyle of being in a brine seawater interface. And we also identified a couple of genes that are directly relevant for biotechnological purposes, namely its capability to produce bioplastics, or its capability to produce enzymes that detoxify heavy metals, which is understandable given that it comes from an environment where it's exposed to high concentrations of heavy metals. So these can be useful in cleaning up the environment, cleaning up contaminated sites. For me, the most interesting microbe that we ever isolated from deep sea brines is this one, haloplasma. And so if you are a microbiologist, if you've had any sort of prior contact with microbiology, and even if you haven't, you're probably aware of this, from a morphological perspective, microbes tend not to be very diverse, okay? They normally have one of two basic shapes. They're either spheres or they're rods. They look a little bit like big cigars or something. And so whenever you find an organism that doesn't fit this type of morphology, it is quite a big deal. And this is the case for haloplasma. So haloplasma has this sort of central round body, but the unusual part is that it has these tentacles. And the tentacles, I'm going to show the video again, the tentacles alternate between this linear conformation and this helical coiled one. So the tentacles contract and relax, which is something that is unique. This has never been seen in other organisms, other microbes. So it does have a unique morphology, very unusual cell contraction cycle. It does have genes, so we also sequenced the genome, and we know that it has genes for cytoskeleton elements, which might be important for biophysics studies. And we're still trying to understand this microbe. There are a lot of really interesting questions that we're asking about it. And so we did some scanning electron microscopy studies, and we got a little bit more additional elements to this unusual morphology. And from a phylogenetic perspective, when we looked at the DNA sequence of this microbe, we were able to check, to confirm that it was a completely new group of microbes, a new lineage that had this really interesting placement between two big groups of microbes, the firmicutes and the mollicutes. So it might be some sort of missing link between both, because it does share some similarities with both groups. Haloplasma is a new order at this stage, and it likely represents a new phylum. We need to get more additional microbes to be able to confirm this. So it is quite a big find, big discovery there. A couple of additional studies there. Based on metagenomics, so the analysis of total DNA content from the environment, we were able to get first insights into viral diversity. So there are also viruses in these environments. And there's some high levels of novelty. So there are a lot of new viruses down there. And this was particularly pronounced apparently at Atlantis 2 deep, where conditions were most extreme. We also were able to confirm that similarly to what is observed for microbes, viral communities are also stratified across the brine seawater interface. So the communities that you see higher up in the interface are very different from what you get just 1 meter below it or a little bit below it. An additional highlight of our studies, and this is directly linked with the ROV mission that I mentioned earlier in this talk, is the discovery of a new type, mussel beds at the brine seawater interface at this sort of beach area of one of the brines. We were able to collect some samples from there, and we collaborated with some experts in bivalve studies. And so we described this as a new genus of mussels, so completely new to science also. So it's not just microbes and viruses, you also have macrofauna there, and new animals, new more complex organisms living in these type of environments. There are also some additional studies that were done on bioprospection and looking specifically at applications, but I won't really go into this now. And a final highlight that I wanted to mention here, which was something really unexpected to me, was the accidental, well, it was a couple of plates that I had in an incubator. I sort of forgot them for a little bit longer than I wanted to. And after I came back from holidays, I realized that I'd forgotten them there, and I realized that the colonies look different. And so the background that you see there in each one of these pictures is the surface of individual colonies. And then I saw that they had these weird circular structures on top of it that I didn't really know what it was. We were able to extract them, clean them, and analyze them. We now know that they are biominerals, which are really interesting things, which leads us to the second part of the talk, biominerals. So the precipitation of minerals by living organisms, so the production of biominerals, is something that has been reported. Like, people know we have several animals that are able to produce shells. Interestingly, there's a huge amount of microbes that also are able to do this. They're able to control or induce the precipitation of different types of minerals. We are specifically interested, and it started with that accidental discovery that I got more into this, more into geomicrobiology and biominerals. We were specifically interested in microbially induced calcite precipitation, so the production of carbonate biominerals. There's a wide variety of things that are involved in this, and while I need to acknowledge Philippa Loftus, my former master's student who just recently finished. Part of the work that I'm covering in this section is directly linked to her thesis and her work. And so why are carbonate biominerals interesting? Well, they've been receiving increased attention. Part of it has to do with the fact that they have very wide range of applications. And so they're already used for a variety of different things. Most important one is repairing things. So carbonate biominerals are ideal filler material, and so they're used for self-healing concrete, for example. They're used to restore old buildings, to restore work of arts like statues, for example, and they're very, very useful for this type of thing. They're also used for improving quality of soils for agriculture and cleaning up the environment. They can also be used for bioremediation. One of the most recent and most interesting applications, though, is its potential use for CO2 sequestration and helping us to fight and control climate change. The microbe that is most commonly used for applications, and that's has been more heavily studied, is Bacillus. And so that's the reason why we focus specifically on this genus of microbes. Bacillus is a very well-known producer of carbonate biominerals. There's a huge amount of species with different capabilities. There's a very big data set, so we have access to genomic data, and they're relatively easy to grow. So it's a really handy microbe group of microbes to study this process further. So despite the wide range of applications that I mentioned here, and the fact that people have been using this for some time now and have been using this extensively and for more and more applications, there's still a lot that we don't know. We don't really understand much of why is it that microbes produce these minerals, or how is it that they're producing it. As first part of this work, we were identifying genes that have been reported as linked to the capability of producing biominerals. And so we came up with this list of genes, and we were interested in developing a new approach, a new tool, a new technique to quickly identify new producers of biominerals. And so we looked at several different type strains, several different species of Bacillus. We analyzed the genome, we analyzed all of the genomes that are publicly available. And here you see, it's difficult to visualize, but you have here in this square, you have the results of this analysis, this cross comparison. And it's maybe a little bit easier for you to see here. This is a split phylogenetic tree that shows several different species of bacillus. Underlined in red are species that have never been tested for carbon by mineral production, and underlined in green are the ones that we know that produce it, that have been reported before in the past. Here you also have a key that shows the several different genes that we've identified from literature search as being implicated in this process. And so here are some of the highlights of Philippa's work. We were able to map and identify this sort of genomic fingerprint associated with carbon by mineral production. And we were using this as a way of quickly identifying specific clusters and looking for microbes that might be good producers of carbon by minerals. This was followed by work done in the lab. And so we incubated several different species that we knew that produced it, and species that we'd never been tested. And we followed the growth of the colonies across 21 days with regular pictures that were taken to record what was happening. And we see an increase in the number of carbon by minerals with incubation time, which is expectable. Okay, so we do see, and I'm not going to spend a long time on this, we do see some interesting trends we do see some interesting phylogenetic trends on the morphology of minerals. It seems that species that are closely related tend to produce minerals that are more, that resemble each other most when you look at other groups, when you compare them with other groups. And this is really interesting. This is really interesting insights for applications and application-driven research. We were also able to identify, we also looked at yield, how much biominerals actually produced. And we compared with some species that are heavily used in biotechnology, and we had some really interesting results there. To summarize things, we were able to develop this new tool based on genomic analysis to quickly find new biomineral producers. We have successfully identified already 10 new species of bacillus that are able to do it, and we had no idea, nobody had ever tested them before. We do see some interesting trends and correlations that we're studying further, and we've identified some new relevant producers that might be useful for biotechnology. But this is part of ongoing work, so this is just the first step of this research. And geomicrobiology is a really interesting field. It bridges the gap between biosciences and geosciences, and this sort of in-between area, and areas that were originally thought of being completely separate can actually be linked together, actually connected via microbes. Which leads us to the third and final part of this talk, exooceans, and a few other things. So the microbiology by default tends to be very cross-disciplinary science, and I mentioned geomicrobiology before. The most extreme case of this cross-disciplinarity of microbiology is its link with astrobiology. And of course, well, I mentioned before this human-centric perspective that people used to have and sometimes still have, but it's slowly changing. People used to think that we were the center of everything, that we were special, that the earth was the center of everything, that we were exceptional, that we were unique. And the thing is, we're moving away from this sort of perception. And so we're looking for evidence of existing life or past life in other places in the solar system. And of course, a big part of this discussion has traditionally been centered on Mars. And this is linked with the history of the observations of Mars, namely the works of Schiaparelli and Percival Lowell, and the drawings that they had and the interpretation that they did seemed to imply that there was some sort of lost civilization on the surface of the red planet, which is something that, of course, now we know isn't really the case. And this fascination with Mars has fueled a wide variety of different pop culture phenomena, both in literature and movies, past, present, and the future, definitely, and even in music. And so, of course, as I mentioned, Mars is becoming increasingly interesting and relevant. There are 3 new missions that are going to be launched this month alone. So it's currently a quite hot topic, but this is not what I want to talk about today. I want to talk about the icy moons. So several icy moons of the outer solar system have been getting increased attention also. More particularly, Europa and Enceladus are now seen as very, very interesting from an astrobiological perspective. And this is linked with evidence for global scale oceans. There's still a lot of unknowns at this stage, so they are cold. We do know that these oceans are briny, so they have high salt content. This is something that we already know. And so there's still a lot of unknowns, both for Europa and Enceladus, depending on the model that you use and depending on the discovery of the further exploration and new data that we get from there. We will get a better perception of what exact conditions can we expect to be existing in these exo-oceans. Some models point to alkaline brines associated with sodium and carbonate. Some point to acidic brine dominated by sulfuric acid, or neutral brines that might have high magnesium content and sulfate. We do have some high pressure conditions, and we do have localized hydrothermal activity in higher temperatures. And this is the case both for Europa and of course for Enceladus. Enceladus has this sort of visually striking plumes that come out of the South Pole that have become quite famous. But of course, getting samples from these types of environments will take several decades still. So to understand and to study these type of environments, to study these exo-oceans, we need to focus on our own planet. And so our planet has several different extreme conditions, has a wide range of different conditions. And if we map the range of environmental conditions on our planet and where we can find life, and do similar efforts with other locations in our solar system, we might actually see that what we originally perceived as extreme conditions on Mars or Venus or Europa are actually not that extreme and might actually be quite welcoming for some of the microbial life forms that we have on our own planet. There's a variety of different terrestrial analog sites, locations that have conditions similar to the ones that you find in other planets. So they've been mapped, they're studied for a variety of different purposes. And we keep on finding and identifying and flagging new additional ones. Regarding the specific case of terrestrial analogs for exo-oceans, it's a little bit complicated because it's not always easy to find locations where you have high pressure, hydrothermal input, high salinity, and so brines with a varying degree of chemistry with different conditions. Luckily for us, deep sea brines do provide these sort of conditions. They do provide quite a wide diversity, and so they're ideal testing grounds. Regarding future research, deep sea brines provide this ideal testing ground for testing the limits of life, a source of microbial strains for testing and exposure experiments, long-term viability and preservation, and also to develop and test new technology that is useful for upcoming missions. I will skip the next session on planetary protection, but I might get back to it for the Q&A session if people want to know a little bit more about this, because I just checked the time and we're running out of time here. So I'll just skip these slides here. So I just wanted to finalize with, we started with this perception that microbes are everywhere and we live in a microbial world. It could be that we are moving to a scenario where microbes might actually be everywhere and we might be present in a variety of other locations in our solar system. And so microbiology and astrobiology is providing the study of brines and halophiles is providing vital insights into answering the biggest question that humanity has ever asked. Are we alone in the universe or not? Okay, so that's it for the talk. I just wanted to acknowledge several different institutions institutions where I worked and I've collaborated with, as well as funding agencies throughout the years. Thank you once again for attending. Feel free to contact me, look me up on ResearchGate, on Twitter. You can send me an email if you're shy about asking questions in public, or you're interested in knowing more about the research that I mentioned here today, or about the State Key Lab in general, or the opportunities that we have over here. And so I just leave it in the final slide. I want to mention that next week's webinar, as part of this current seminar series on astrobiology that involves several different research centers across the region. We'll have Sid, and you know him from earlier today. He was the guy that was introducing me. He's the one that will be presenting, be giving a talk next week. Thank you very much once again. I'm happy to answer questions.
Thanks a lot, Andre, and thanks for the heads up for my talk. So do we have a hard stop, Andre, at your end?
No, it's flexible. I'm flexible. If people are questions, I'm happy to answer them.
Okay. I see Maggie has raised her hand. Maggie, do you want to come in?
No, it was just like clapping.
Oh, okay.
Yeah.
Sorry.
No worries. Maybe you attend to the chat room, because I saw quite a number of questions have came before me.
Sure. So, Andre, is it okay? I'll just read out the question to you.
Yeah, that's fine.
Okay. So we have a question from Anurup Mohanty. He asks, in general, how slow are the life cycles of extremophiles which thrive in deep sea brines?
Okay, that's an excellent question. Very variable. It is one of the challenges of working with certain types of environments and certain types of microbes. And it does explain some of the issues with the plate count anomaly. Some microbes don't grow on plates, and we don't see them on plates because it just takes them very, very long time. Regarding microbes specifically on the deep sea brines, it's variable. They're slower than your standard microbes like E. coli. E. coli grows super fast. Bacillus grows really fast. Extremophiles in general tend to have slower growth rates. It's still manageable, but I did have variability. So for example, Salinisfera grows in 2 days, for example, and Haloplasma usually takes around a week to a week and a half. I did have friends in the lab in Germany that were working with cold environments. When you're working with cold environments, it tends to be a little bit trickier. They had a microbe that took several months to grow. And so it was something, and it was a friend of mine that he was doing his master's, and he wasn't able to continue working with that microbe because the microbe takes several months to grow. Then you're unable to do any proper work with it during the duration of a master project. It is one of the challenges of working with certain types of microbes. Thank you for the excellent question.
Awesome. So there are a few questions. I'm trying to keep it sequential. Let's see. There's a question, does the deep brines exploration cause any disruption to the marine life?
No. Well, no. So exploring these brine locations is not disruptive. You did see the ROV sort of disrupting that sort of biofilm structure, but it formed again, and there were multiple patches there. So regarding microbes, it's something that we normally don't worry about that much. Microbes are quite resilient. The techniques that we use for sampling tend to be the least disruptive possible. Regarding possible effects and disruption to regular marine life, brines work as a sort of a separate body. They don't really mix with seawater that much. There were some concerns, though. There was this project from Saudi Arabia, and it's been discussed for many years now. I hope that they never go ahead with it. There are some plans to do some mining in Atlantis too, because you have accumulation of several different types of metals. Some of them are economically very interesting, so they're worth a lot of money. And so there have been plans to try and extract metals from deep sea conditions, namely from this deep sea brine. And that might create bigger environmental issues for regular organisms living in the Red Sea.
Thanks. That question was from Kaustubh Rajesh Gupta. Here's a question from Hiroki Nishimura. After fixation of the cell, is it possible to extract single cell genome from the cell?
Okay.
I'm not sure.
Well, that's an interesting question. I think it should be viable. I do know that for single cell genomics, you normally rely on cell sorting techniques where you try and get individual cells or a small number of cells into wells and you do it. So theoretically, you could do it. That should be feasible. The advantage of this technique, so the optical tweezers is not necessarily the best suited technique for single cell genomics because you have other more high throughput techniques to do this. The advantage of using the optical tweezer is that it will allow you to get pure cultures of these microbes. So you can get more biomass and you can actually do more studies with it. That's the big advantage there.
Another connected question is from Sudipta Biswas. Doesn't the laser used in the optical tweezer method disrupt the cellular components of the microbes?
Another great question. Well, one of the tricks of using the optical tweezers is you don't keep the laser on for a long period of time. So it's normally, the perception that we have is that the laser has a detrimental effect on cells. And so some cells will lose viability from it. So normally when you're using the optical tweezers and you're interested in a specific morphology, imagine that you're looking for long rods, for example. You normally pick one individual one and you inoculate one tube with it, with that individual cell. And then from the same culture, you repeat that multiple times so that you increase your likelihood of success. Because success rate is not 100%. It's not each individual cell that we pick that will grow and will do well. And this could be a combination of factors. When you're picking individual cells, you don't know how healthy they are. It could be that they're already at a later stage of their life and could be partly damaged. It could be some damage that might come from the laser, but the success rate is usually quite high. But we do tend to do it fast. We do it as quickly as feasible to minimize any sort of potential detrimental effect that it might have on cells.
Awesome. Question from Sky. Did you say the viruses are more prevalent in the harsher environments? And if that is the case, then why is that so?
Okay, I actually didn't say that.
Yeah.
I didn't mention that. We do see, that is commonly the case though. Viruses tend to be very, very abundant in extreme environments. And one of the reasons why, one of the explanations for this is lack of other forms of predation. So if you're dealing with less extreme environments, you tend to have more complex trophic networks where you have different types of organisms eating up bacteria and feeding on them. When you go to extreme conditions, diversity tends to be reduced and more complex life forms tend to eventually vanish or be heavily reduced. So it seems that viruses seem to play a stronger role in controlling microbial populations. So they play a very important ecological role there.
Awesome. There's a question from Sky also about the biomineral production that you spoke about. Mm-hmm. Do you have an idea of the efficiency scale of carbon sequestration that can be achieved from biomineral production?
I don't have exact numbers on this. I do know that that this is a relatively new technology and that there were some pilot projects that were looking specifically into this. So my belief is that this is a dynamic field and this is something that is changing and is shifting, but it's something that has been getting a lot of attention.
Okay. I think I had skipped one question, so I will get to that. This was from Annalise Meyer. She asks, could it be that an iron-rich particle accumulation effect from redox transitions of different ion states if you're getting oxygen concentration changes across the density interface?
Yes, I guess that this question has to do with the red patches.
Mm-hmm.
Well, the truth is, we don't really know. At this stage, we don't know what it is. This is another possibility, yes. It's something that we considered. It doesn't seem to be the most likely scenario, given the pattern of disruption. When we approach with the ROV, it seems like it's a biofilm-like structure that is being disrupted, rather than something geochemically generated. It's something that we're planning on investigating further. We've received green light for an expedition to the brines of the Red Sea. We're just waiting for things to be in place, and we're expecting to go back to this location possibly by the end of next year. We'll see how coronavirus affects the plan, but we're planning on getting back there and specifically sampling the red patches. And we'll be able to answer that question and know exactly what it is and why is it there.
Awesome. Another question from Hongyan Zhou about the tool of the genomic-based analysis to find new biomineral producers. How efficient or reliable is it, since microbes adapt so fast depending on the environment?
Okay, it is very reliable. So all of the work that we have done has been done with type strains, and type strains are like examples, they're like preserved copies of what is, they're like preserved copies of what a species is like. They're the reference when you're comparing different species. They are kept in culture collections, and they are preserved in the state that they're in. So regarding analysis of genome and testing in the lab, it is quite reliable, and it should be a stable feature.
Question from Shaolin Lee. Do you think other liquids can replace water for extraterrestrial life?
Well, there's big discussions on this on a regular basis. Yes, there are other possibilities. It's at this stage, it's more of a theoretical discussion, as in it's feasible that other liquids could replace water. It still seems that water would be the preferred liquid to support life. Of course, we always need to be a little bit careful with our expectations, and we are limited by the fact that we only have one example. We only know life on one planet. And so we're a little bit limited in our understanding of how life can appear and how flexible can it be. And the truth is, when you're discussing extremophiles and looking at extreme environments and some of the amazing microbes that you see from these sites, if you would have shown it to microbiologists just a few decades ago, they would have thought that they were alien creatures somehow, or that they were doing things that were impossible. So it's something that it's a speculation at this stage, and it's mostly geared towards the theoretical field.
Okay, here's a question from Kaushal Yanchandani. He asks, what are the features that you use to classify an extremophile in the brines as alive or active? Do the extremophiles go into a state of slower metabolism or hibernation, given that they live in extreme environments, which might be nutrient deficit during certain seasonal cycles?
Okay, there are a variety of techniques that can be used. So when you're using microscopy, you can use specific stains. You can stain cells selectively so that you're able to see which cells are active and which ones are, well, which cells are alive and which cells are not alive. Regarding dormancy and slower metabolism, well, it's known that microbes can have long-term viability. Several microbes that live in deeper geological layers, for example, are very slow. They have very slow metabolism, and they're able to survive long periods of stasis of sorts. High salinity environments in particular are an interesting example of that. It seems that microbes trapped in salt crystals are viable for an incredibly long period of time, which is something that is really interesting and really relevant. Some of the studies that have been done have been subject to heated debate whether they're fully accurate or not. but there's direct implications on the search for life on Mars. So even if we don't have active life on Mars nowadays, if we look at salt deposits over there, then salt would have acted similarly to what it happens on our own planet. It could have acted as a way of preserving living organisms and any type of biomolecules that can be used as biosignatures.
Thanks. So the question from Kashish is, what if we find a microbial extraterrestrial life? What happens next?
Okay, I think that would be a massive discovery. That would probably be the biggest discovery ever. If we have space mission that goes to Mars and suddenly we find some sort of microbe living there, there are two big alternative scenarios. Option one, it is similar to life on our planet. It shares several common features, and it wouldn't be too unexpected. It's known that there's been intensive traffic of material between Mars and Earth. And so we could have had traffic of molecules that could have helped start life. And we could have even had traffic of microbes from one planet to the other. So if we find life on Mars, for example, and it's similar to our own, it might fit within the tree of life. And that would be a very big change to our perception of where life started. Maybe life didn't start on Earth, maybe it started on Mars, and the implications would be very big. The other alternative, of course, is that we find life on Mars and it's completely different. That's even more interesting because it would give us a second example, a second tree of life, and it would give us a better idea of how life is able to originate and evolve and adapt and thrive in different locations across the universe. So I think both scenarios are really, really interesting.
Another question from Anurup. He asks, piezophiles or halophiles for deep sea brines are found in extreme depths of seas on Earth, and they're not exposed to radiation, I suppose, but Europa receives high levels of radiation from Jupiter. Do you think that can be a challenge for life if it exists there? And I'll also connect another question about the low temperatures. Could that also be a challenge for the origin of life?
Okay, yes. So all types of extreme tend to be challenging, and the combination of multiple extremes is an additional challenge. It's one of the reasons why it's so important for us to study extremophiles, study extreme environments, particularly the type of environments where there is a combination of different types of extremes. Okay, regarding low temperatures specifically, lower temperatures will reduce the speed of chemical reactions. And what this implies is that life would take longer to start and it would take longer to evolve. So if you were looking at very cold environments, it makes it less likely for life to be found or for life to evolve. Regarding specifically the situation with the icy moons, exo-oceans, well, the expectation is that there is some sort of hydrothermal activity associated with these exo-oceans. So you would have locations where conditions are different, where temperatures are higher. And so you would have, it's not that the entire ocean will have exact same conditions. There would be variability. So I think that there would be a lot of interesting things to look into.
Awesome. So there's another question about the slides. Would they be available to the participants for reference at a later point?
Well, I think that Maggie might be able to add some information there. I believe that she's recording this talk and we'd be releasing it publicly so that if other people that missed part of it or want to check it again, or people that weren't able to participate, they will be able to check it.
Okay, yes, so yeah, Maggie confirms that the chat is being recorded. Okay, so there is a question about the chemical formula for manganite from Yangzhang Li. He asks whether it's, I don't know if you can read the question.
Yes, I can.
Yeah.
I'm not really able to answer this specific question though, I'm sorry. Yeah, it could be that it's a typo from my side. This is a photo that I took in one of the expeditions that I had. And this was the information that I received from the local geochemist. And he said, yeah, there was manganite, manganese oxides in one of the samples. This is the formula that he gave me. Could be that it's a typo from my side.
All right. So question from Sri Kavya Peneru. Could you explain why the phylogeny of these brines were far apart? There should be some ancestor in common.
Well, there is some ancestor in common. So they still fit in the tree of life. The fact is because conditions in the brines are so different and they're so separate from regular seawater, this has led to the development of new groups of microbes that are not really directly linked with microbes that we find elsewhere. So that's the reason why they're brand new phylogenetic groups.
Mm-hmm. Then this is a question I think you've gotten a lot before. So what happens if what if ET life is vastly different from Earth? And would that limit our ability to look for this life?
Yes, yeah, well, it's a big issue, and it's something that is discussed more on a theoretical level, as I was mentioning before, for example, with alternatives to water as a liquid, and trying to push the boundaries a little bit. We are limited by the fact that we only have the one example. And it's something that we need to be able to think outside the box and think of other alternatives and try to fight our own biases when we're looking into this. But it's not always easy. So it is challenging.
I just have, oh, okay, more questions coming in. There's one that's a bit broad. I don't know how you will take that, but I will still throw it towards you. It asks, how do geological studies help in astrobiology, both in the search of extraterrestrial life and terraforming any other planet?
Okay, well, geological studies are, of course, very important as geology and geochemistry limit and condition life. They're directly linked. And I mentioned one aspect. I was talking about biominerals, but there's a huge ways in which microbes interact, condition, and change global geochemical cycles. So they're both factors are intimately linked. So one field is vital to the other. Regarding terraforming of other planets is something that we need to be a little bit careful with, particularly as at this stage, we don't know if there's any sort of life on Mars. Any sort of terraforming event would, of course, have implications. So we need to be careful not to be wiping out entire ecosystems before we even know that they're there. Well, extremophiles in general and microbes would be a useful tool in terraforming opportunities, but it's something that we need to be careful, and we're not at that stage just yet.
Here's an interesting question, and I'll morph it a little bit with the permission of the person who's asked this question. She asks, what are the measures taken to protect intrinsic value and protect their niche while exploring and bioprospecting analog sites and planets? I think I'm also curious to know about when you do these deep sea expeditions, are there some specific permits that you need to take and how different is it in different parts of the world?
Yes, well, we need special permits. All of the samples we collected were in Saudi waters. Then there are a couple of brines that are sort of in this gray area where it's not really clear if it's Saudi, if it's Egyptian, or if it belongs to Sudan. So you normally just need to get permits from all three. The common thing, and I've had this experience in several different countries regarding sampling, the easier thing is have a local partner. If you have a local partner and if you're engaging the local researcher community, it makes things much easier. I'm not a fan of researchers coming from big research centers with lots of resources and going to a developing country and just sampling everything and going back home and not linking with the local communities. I can give as an example, I'm heavily engaged with research activities in Cape Verde. It's a place where I worked, I had a faculty position there. Cape Verde is a small island state off the coast of Africa, more or less in front of Senegal. We are collaborating with researchers there. We've sampled several high salinity environments there, and we actively collaborate with them. So rather than just having this sort of exploratory view that you just go get samples and go back home, you engage and you're involved with the local researchers, and you're linked with the local authorities so that local populations, if there are any sort of benefits or if there are any sort of concerns regarding protecting threatened environments, that local authorities and national governments are aware of this, are aware of the value that is associated with these sites, and that facilitates their protection.
Yes, great answer, André. I completely echo your thoughts that we should be working closely with communities and research institutions that are close to these analog sites. And I think this society that we formed is working in that direction to enable those collaborations. Another question from Anurup: do we see multicellular organisms in deep sea brines?
Okay, that's well, I did mention the mussels that we found, for example, in one of the deep sea brines. Inside the brines, conditions are apparently too harsh for more complex organisms. But at the brine seawater interface level, we do see several different multicellular organisms. We even see fish, we see shrimps that seem to feed on the high particle load that you see there. Every once in a while, you find some sort of pickled fish that probably dove too much into the brine seawater interface, and it's just there pickled. Yeah, we found, we find that sort of thing.
Mm-hmm. Question from Maggie. Are there any common organisms that you have found in all these different deep sea environments or salty environments? For example, the deep sea brines, the in permafrost brines, and in man-made salt pans. Are there any common organisms?
Yes, there are some commonalities. Members of the archaea tend to be the dominant life forms in high salinity environments. It doesn't seem to be necessarily the case with deep sea brines. We do see some groups of microbes that are present in deep sea brines and also in more other types of high salinity environments. Of course, the more interesting ones are the ones that are specific to the deep sea brines and we find nowhere else.
Mm-hmm. I think I have asked all the questions. Maybe we should encourage people, they would like to ask questions by unmuting the mic. If there's any questions anybody has.
Well, if not, I'm fine. As I said, I'm fine with doing follow-up answering via email. People found specific things that they're interested in. I'm fine in continuing the discussion online.
And you also mentioned career opportunities, so they should write to you directly in case if they're interested people? Yeah,
it's easier. Even if it's not linked with astrobiology, it could be that we have, I'm assuming that we have members here in the audience that have some interest in astrobiology, but it's not necessarily their research field or research interest. So if we have students, people looking for postdocs or people looking for faculty positions, we have a wide range of different research fields in the center. So I can direct people to the right contact person.
Awesome.
Okay.
So I think with that, we can close today's talk. So thank you very much, André, for giving this talk. I really hope I enjoyed, and I absolutely loved that video that you showed with the bright red biofilm layer. It was really exciting. Yeah. And it was very informative. The way you answered the questions was very well done. So with that, we will close today's session. And I will be taking up next week's session on an overview of activities in India and in perspective with my newly formed center, which is just a year old. But I'll talk about some of the different things we have kickstarted. So with that, thank you, everybody, and look forward to seeing you next week.
Yeah, also very excited to see it next week.
Awesome. Thanks a lot.
Thank you once again for all for attending. And if you're interested in knowing more about the activities of the Astrobiology Society of Asia Pacific, go to astrobiology.asia and check. There's you can sign up to receive regular updates and you can participate in the discussion forum.
Awesome. All right. Thank you very much.
Thank you.
Take care.
Bye-bye.
Yeah, thanks a lot, everyone. Thanks a lot, André and Sid.
Thanks, Tony.
Thank you.